Hello, everyone. Thank you for joining us today. This is the second seminar for the Spring 24 BERD series. Today we have, just before we start the talk, I wanted to remind everyone that the Zoom session will be recorded automatically. Right now, all of you are muted except for the co-host. If you have questions during the talk, please free to type the questions in the chat window, and provided the speaker is okay, you can unmute yourself and ask the questions as well. For the registered participants, we will share the slides after the talk with a short feedback survey. So today we have Professor Xian Xu from Penn State Statistics. He's an assistant professor of statistics here at Penn State. His research work mainly involves developing statistical and computational methods for large-scale genetic and genomic data. He has a long list of projects and publications, some of which we might hear today. So without further ado, Xian, take it away.
Okay, thank you, Pramani. Let me share screen. Okay, let me do the presentation mode. So Prabhani, can you help me confirm if you can see my slides and also hear me talking?
Yeah.
Okay.
Yeah. We can see the
great. Thank you. Thanks everyone for coming to this virtual talk, and also thanks Mosok. I'm not sure if she's here today or not, but thanks for her invitation, and also thank you very much, Prabhani, for the very nice introduction.
Well, you are most welcome, Sean.
Oh, you're here. Hi. Okay.
Thank you for giving the talk.
Okay, thank you so much. Also, thanks Prabhani for helping me set up the slides and all this logistics stuff. Today, I'm very humbled and also grateful for this opportunity to present some recent, some very new work at the interface of Bayesian statistics and human genetics. I think this may have, of some interest to both statisticians and geneticists in our Penn State community. So here's a brief summary of my talk today. I will start with some background introduction about a very useful approach in human genetics nowadays called the Genome-wide Association Study, or GWAS. And then I will present a Bayesian statistical framework that I've been developing for many, many years. I'm still working within this framework called Regression with Summary Statistics. After that, I will demonstrate the real-world utility of this framework in 4 different applications in GWAS. And then I will conclude today's talk with several ongoing expansion of this framework just to address the emerging issue of summary data coming from GWAS these days. So some of you in the audience may know this. Over the past 2 decades, GWAS, or Genome-wide Association Study, has been a very useful tool to identify common genetic variation underlying common and complex human disease and trait. Despite being a very powerful approach, GWAS has a surprisingly simple study design. So all we have to do is to recruit a large number of ideally unrelated individuals, measure their DNA and some phenotype information, and then perform statistical analysis to identify significant associations between genetic variation and the variation in human health and disease. Let me give you more introduction about the actual data we're working with in a typical GWAS. So GWAS consists of 2 components, one called genotype and the other called phenotype. So genotypes are essentially measurement of some kind of genetic variation across different individuals in the study cohort. So for today's talk, I will largely focus on one type of genetic variation known as single nucleotide polymorphism, or SNP, which can be coded as a data between 0 and 2. So in this box here, you can see 4 different orange boxes in the same location of the genome sequence. So this is an example illustrating one SNP across 4 different individuals. So as you can see, even from this simple example, there's some variation going on between different individuals. For the phenotype data, so it's that kind of data just are are some kind of characteristics of our human beings. It can be some binary data indicating whether that individual has certain disease or not, or it can be some continuous measurement indicating, let's say, body mass index for obesity or blood lipid level for cardiovascular risk. Once we have this genotype and phenotype data available, the ultimate goal in GWAS is to understand how does genetic variation affect phenotypic variation. So let's formalize this introduction in a mathematical language because this is where I come from. So for any typical GWAS data, we can organize them as a Y vector, which is an n-length vector of the phenotype individual of the phenotype information from n different individuals. And then the genotype data can be organized as an n by p matrix, where n indicates the number of individuals again, and p indicates the number of SNPs we analyze in a study. So typically N range from 10 to 500,000 these days, and P range from 1 to 50 million. So if you're looking for a big data example, GWAS is a very good place to start with. So once we have this large amount of data available, the standard analysis in GWAS, even up until now, is to perform single SNP association testing by simply regressing the Y vector on each column of the X matrix, and we can further summarize the result as this B hat J, the marginal effect size for SNP J, the sigma hat J, which is the standard error for the marginal effect size estimate, and also the PJ, which is the p-value for the marginal effect size estimate. And then GWAS publication will usually release these 3 summary statistics for each SNP, whether it's significant or not. They just release all the results as part of the publication process. So whenever I'm presenting this kind of data, especially to statisticians, the very first question I got from the audience is that, okay, now you have your data nicely organized as a Y vector and an X matrix, and why you're okay with doing this kind of simple linear regression type of analysis rather than doing more complicated multiple regression analysis. So actually, I'm with them. So indeed, the multiple regression model is a more reasonable model to analyze this type of data, because this multiple regression model help us capture the polygenicity that multiple genes can affect a complex trait such as human height or human skin color. So here's just a very simple visualization illustrating that different genes can work together to regulate the variation of human skin color. And then for human height, there's like hundreds of genes known to us these days can do the job, okay? So conceptually speaking, multiple regression is indeed a better model to consider to analyze this kind of GWAS data. However, and also, we've learned a lot from our traditional regression class in, here at Penn State or in any other institutions. We teach a lot about multiple regression, and we have frequentist approach by, by minimizing the distance and introducing some penalty to deal with the high dimensional case. And we also have the Bayesian approach by obtaining the posterior distribution for the regression coefficient given the data we observe. However, whether regardless what kind of regression approach we're looking at here, we're actually making a very important assumption without saying it, and that is we are assuming we have the individual level data X and Y available in the first place before we can conduct any multiple regression analysis. So the problem is that, especially in the field of GWAS these days, getting access to the individual level data is very difficult, nearly impossible. And here's one example illustrating the difficulty of getting the individual level data. So so over the past few years, I have the privilege of working with a large group of people called this Global Lipid Genetics Consortium. So this consortium consists of 201 different cohorts involving 1.65 million individuals across the globe. And this table just shows you the sample size breakdown by the ancestry group, because we're looking at the entire human population from each continent. So due to the privacy and regulation issues, nobody in this consortium can ever, combine the individual phenotype Y vectors and also individual genotype matrices X from all 201 cohort and to form that huge Y and X data matrices for us to perform the standard multiple regression analysis. So this is just one example, and there are many other consortium examples. Just keep telling the same story. Putting this large data matrix together is logistically impossible and even legally impossible, because for certain countries, their genetic data can never leave their server located in within their country borders. Okay, so since now we have this challenge of not being able to get the individual level data and do our traditional multiple regression analysis, we have to find a different way out, because we really like the multiple regression model because it provides a better description of how genetics affect disease and health. So thanks to the NIH's decision back in 2018 to broaden the public sharing of genomic summary result, the GWAS summary statistic produced by the single SNP association testing procedure I mentioned earlier are increasingly available to the entire public these days. And then you can see the trend from this bar plot here, indicating this type of summary data are getting more and more available in the public domains. So this kind of increasing and widespread availability of summary data give us an idea. So because I'm trained as a Bayesian statistician, I'm going to start with the Bayesian regression. So the fundamental of Bayesian statistics is this so-called Bayes theorem that is here. If we want to derive the posterior distribution of the regression coefficient, I only need to combine the regression likelihood with the prior. And so the prior part has nothing to do with what kind of data you're working with, because this entirely comes from our prior belief without any data involved. So the only part that depends on data is the likelihood. So now we cannot use the traditional regression likelihood introduced in our textbook where X and Y are required. So that motivates us to develop a new likelihood that does not rely on X and Y, but instead relies on the summary result that are published together with GWAS these days. So this is our solution. In short, our solution is this so-called regression with summary statistic likelihood, or RSS likelihood. So in order to derive this likelihood, we just need to make very standard and simple assumption about how GWAS data X and Y are generated in the first place. And then the key assumption is actually here. So we are assuming that for each individual, their phenotype data is generated from the genotype data under the standard regression model, multiple regression model. And then this beta vector is the multiple regression coefficient, okay? And in genetics, this beta vector tells us the true effect size of each SNP on the target GWAS phenotype we're looking at here. And then based on this and also this assumption is a standard in a way that this is the same as assumption people nowadays are making to describe the polygenic architecture of complex human trait. So we're not doing anything that is significantly different from the rest of the field. So under these very standard assumptions, we can use some asymptotic tools from statistics I will show later on to derive this p-dimensional Gaussian distribution as our RSS likelihood. So this is a p-dimensional Gaussian distribution where p is the number of SNPs considered in this GWAS. And then the parameter of interest for this likelihood function is this beta vector, which is the same beta vector that we used earlier to assume how does the data come from. Essentially, this beta tells us the true effect size of each SNP on the disease for a given GWAS. And then the input for this data likelihood comes from two sources. So the first source, I use the color green to indicate, and this is the source I mentioned earlier. So this comes from the GWAS summer statistics that are part of the GWAS publication this day, very easy to obtain. And then there's another source come from so I use a different color just to indicate that these come from a different source that is not part of the GWAS publication. So this is a P by P correlation matrix. It has a name in genetics. It's called linkage disequilibrium, or LD. For the audience who are not familiar with population genetic terms, you can just understand this as a correlation matrix between different SNPs. And once again, we have very reliable and high quality database such as 1000 Genome or NORMAD these days to help us estimate this kind of correlation matrix between SNPs. So that means everything in this likelihood function is known to us except the parameter of interest, the multiple regression coefficient. So this is a very brief introduction, or this is a very brief overview of how we actually derive this likelihood from the mathematical perspective. So we start with the asymptotic distribution of beta using some kind of central limit theorem type of argument. So we can show that this kind of this beta hat actually follows a normal distribution, a p-dimensional normal distribution. So the asymptotic mean is actually very simple to work with. However, this asymptotic variance is actually very complicated because of this delta C term. So it's so complicated that I don't even want to put that here because it will take an entire slide just to show the actual form of delta C, where the C actually indicates the marginal correlation between each SNP and the phenotype itself. So if this is just any arbitrary statistical model, then it's very hard to proceed. However, we're working with GWAS data only, so we can leverage some empirical observation in GWAS data, which tell us that this C value in general are very small for the vast majority of SNPs, and they're as small as 0, okay? So that means this is a complicated term here that bothers us, but we can safely drop this term without losing too much information. And this is specifically for GWAS application. I cannot say for sure that the same technique can apply to other application cases. So now that we drop this very complicated term, but it's negligible in GWAS data, so we have this very simple form, okay? So this form has a very simple mean structure and very simple variance-covariance structure, but there's still a practical issue in the way that both S matrix and R matrix are unobserved. They're still considered as parameter. So I did a very simple thing in the end. I just plug in some kind of sample estimate for these two parameters because I believe they're just nuisance parameter. My focus should be still the beta itself. Okay, so this is the entire journey of getting this likelihood function in less than 5 minutes. Okay, so I do want to say something about this LD matrix R hat, which is essentially the correlation matrix between SNPs. So in general, okay, so here we have this p-dimensional normal distribution to work with. So in general, it's not a terrible thing to work with a normal. We love normal. I think especially people in my department were crazy about normal because they have very nice theoretical and application properties. However, in my case, the application based on this normal distribution is not that simple because of the scale of P. So the smallest P I ever get to work with is more than 1 million. So having a 1 million dimension multivariate normal distribution in your calculation is not a simple thing. So we need to leverage some kind of structure in the correlation matrix we're having here. So luckily, the LD matrix has this very nice block banded structure in the sense that the entry of this correlation matrix will only be non-zero if and only if this corresponding two SNPs, they are very close to each other. That's one condition. And also they must be in the same chromosome, okay? So this gives us this very nice block and banded structure. So here I'm showing you an example of this structure. So you can see the deeper color indicates a higher correlation value, and then the white color indicates no correlation at all. And then you can see it forms a kind of like a block. There are like one, two, several blocks you can visually see here. And also we have this kind of banded structure in the sense that if you're looking at SNPs that are very far away from each other, they're just completely independent from each other under this structure. So this structure actually gives us two benefits. So first of all, it saves a lot of memory in the calculation. So we only need to load the coordinates and the value of all these non-zero entry into the memory. And second, it also saves computational time because we don't have to do all the computation for p by p pairs, p by p divided by two pairs. We just need to focus on the pairs that have the non-zero value. So this saves a lot of plus and multiplication, those basic computation in any statistical procedure. Okay, so this is how, so in general, this is still a very difficult model to work with, but we have one more feature about our head that really simplify things a lot. Okay, so now that's just a likelihood for me to model the summary statistic coming from a standard GWAS analysis, but I'm pursuing more here because other than the GWAS summary result itself, which is part of the GWAS result, we also want to combine this kind of result with some other information to derive additional biological insight. But actually, before getting into that, I will illustrate two applications that we can do just based on GWAS data alone without introducing any additional information. And these two applications are based on this exchangeable prior, which is a mixture of point mass 0 and a bunch of normals and mean 0. So I'm calling this exchangeable prior in the sense that for all the SNPs, they have essentially the same prior distribution, and there's no difference among SNPs in terms of their prior. So it's a simpler model to begin with. So the first application focuses on the heritability estimation. So once again, I apologize, another jargon. So heritability is essentially the fraction of the total phenotypic variation that can be explained by the genetic factors. So in mathematical language, it's essentially the ratio of this variance coming from the genetic part in our regression model divided by this total variance of y. Okay, and after a sequence of very straightforward linear algebra operation, we can show that this heritability, which is a function of the beta, that makes sense because beta tell us the genetic effect of each SNP, right? So that determines how genetic that phenotype would be, because if all the beta are 0, there's not a genetic at all, then there's no heritability at all. So this beta, this part, so this heritability is indeed a function of beta, and we can further show that this heritability can be approximated by this very simple form of quadratic form for beta. Okay, and this is how we estimate heritability in practice. So we first develop some kind of algorithm such as Markov-Chain Monte Carlo or mean field variation inference to get a posterior of beta. Okay, and then now you realize that this heritability is a function of beta in this quadratic form. So it's actually very straightforward to induce the corresponding posterior distribution of this beta. Okay, so this is actually a technique we teach in our undergraduate class called the change of variable. Okay, so you have the distribution for one random variable, and you can induce the corresponding distribution after transforming this random variable in a certain way. And once we have this posterior distribution, we can just estimate the Hg squared in a very straight way. You can take a mean, you can take a median, however you prefer. And here's the example of estimating heritability in practice. So we apply this RSS approach to the GWAS of adult human height published in 2014. And then this green curve you're seeing here is the posterior distribution of the heritability estimated by our approach. because these are real data results, we don't have the ground truth. It's not simulation. So we did a second best thing. We just applied the gold standard approach in our field to estimate heritability. And that gold standard, I think even until today, is still the linear mixed modeling approach to estimate heritability directly from the individual level data X and Y. However, when we're doing this kind of analysis, we don't have access to the entire individuals because the total sample size is about 250,000. And then due to privacy protection and regulation issue, we only have access to a very small subset, 6,000. But that's okay. We can still get access and apply the linear mixed modeling approach to estimate heritability. So the orange interval you're seeing here indicates the point estimate here in the middle, and then the 95% confidence interval for this estimate. So two messages here. First of all, very reassuringly, our posterior distribution nicely captured this estimate derived from the gold standard approach based on individual level data, which means that our new approach works as the traditional approach. And then more interesting is that if you look at the shape of our distribution is more narrow, like more concentrated compared to the 95% confidence interval that the traditional approach based on individual level that can provide. So you can see the orange interval is very stretched out, whereas the green curve is more concentrated here in the middle. So this is not surprising at all because when we apply the linear mixed modeling approach to build this 95% confidence interval, we only leverage the information from 6,000 individuals. Okay. However, when we apply the RSS approach to summary data, we can leverage a much larger underlying sample size as large as 250,000. Okay. Due to the larger sample size, it's not surprising at all that we are getting more accurate result by lowering down the level of uncertainty. Essentially, the credible interval is a lot tighter. It's a lot narrower than the corresponding confidence interval. And then another application just based on GWAS data alone using RSS is to predict phenotype from genotype. So in our field, we have a term for this. It's called polygenic risk score. And I will come back to this term later on in the end of the talk. But just think of this as a statistical prediction exercise. So you train your regression model, you obtain some kind of estimate for your regression coefficient, and then you apply this predictive model to the genotype data of a new individual whose data were never used in the model training part, and then you do the prediction. And then once you have the predicted value, you can compare with the actually observed value for this new individual to assess the accuracy. So these are the result of applying RSS and several other prediction approach to two different phenotype. One is body mass index, BMI, and the other is height in two different studies. I think these are from US and these are from Europe. Okay, so from this result, we can see that RSS is actually very competitive in terms of predicting phenotype from genotype directly compared with several popular approach such as LD-PRED and then Bayes-R. So I think in this assessment, we are outperforming LD-PRED slightly across the board, which is really nice because LD-PRED is really a popular approach in the field. And there's one more thing to mention here. I'm not the person who generated this benchmarking result. This is actually an independent validation by others. So all I did here is just to teach this group of people in Australia how to utilize my RSS software, and then they independently evaluate the performance of RSS prediction, and then they publish a paper, and I'm not the author of this paper. That also indicates the independence of this evaluation, okay? So it is really nice to see other people in the other side of the world who can actually utilize our approach and independently demonstrating its performance. So now let me get to the more biologically motivated applications, because earlier I mentioned, okay, RSS can do certain things by just using GWAS data alone without introducing any biological information, but it is a very popular trend in the field of human genetics now to integrate functional data from multiple modalities and to improve our interpretation of the GWAS data. And that's why I'm giving two other applications along this line of trend in our field. So to formulate this kind of biological idea or this kind of scientific pursuit in terms of statistical language, so all I'm doing here is to design a set of prior distribution such that each SNP will have their own prior distribution. Because earlier, the previous two applications essentially assumed that every single SNP have exactly the same prior. Okay, but now I'm introducing variation because different SNPs have different biological function. So in order to introduce this kind of variation, I need to define some kind of functional annotation for each SNP as the foundation. So the functional annotation here is essentially a binary annotation for each SNP-J, indicating whether the SNP-J falls inside any functional region of interest. So AJ equals 1 if yes, and 0 otherwise. And once we have this AJ annotation value defined for each SNP-J, we can start building our prior distribution based on the AJ value. So first of all, we can try to model the prior association probability between SNP-J and the GWAS phenotype as a function of AJ. And then we can also try to model the effect size of the beta J under the condition that beta J is actually associated with the GWAS phenotype as another function of AJ. And we can clearly do both, okay? Just how model goes, right? So you can always build any set of model in a very flexible way. And this is actually the approach we pursue. We're modeling both the probability and the effect size as a function of AJ. So here's how we write down the models. So we begin with this AJ annotation, the same annotation I defined in the previous slide. And then I'm using the spike and slab prior for SNP level effect size beta J. So I can even make this more general by using that finite mixture model I presented earlier, but just that's going to complicate the presentation itself. So I'm going to use the simplest possible scenario where you only have one normal component and one zero component, okay? So this zero component really doesn't tell us much. It just tells us, okay, this SNP-J is not associated with that trait, so it's less interesting. So my entire modeling focus will be now in the first component, okay? So the first component, there are two things that I can control. So the first is the pi j. So pi j is the prior association probability for SNP-J with a given GWAS phenotype. So if pi j equals 1, then that tells me, okay, that SNP-J is going to be associated with the phenotype for sure. So I'm modeling the pi j as a logistic function, as you can see here, and it's a function of AJ. So by writing down this logistic function, I'm actually getting a very nice pattern called enrichment. That is, if SNP-J belongs to some important functional category, and then the corresponding AJ will be 1, and consequently, the corresponding pi j value would be larger than the pi j value for the SNPs that are outside the functional region, okay? So you can see I'm introducing some kind of preference or some kind of favorism, okay? And we're calling this enrichment in genetics. And similarly, I can do the same thing for this sigma j squared. So this basically captures the prior variance for SNP-J that are believed to be associated with the trait. So once again, I'm introducing some kind of enrichment pattern here, but this time I'm using a simple linear model in a way that when SNP-J has some function, AJ equals 1, and then the corresponding sigma j square value would be larger than the sigma j square value for all those SNPs that are not part of those functional regions. Okay, so by writing down these two enrichment model, I accidentally or unaccidentally introduced several more hyperparameters, as you can see here. So this will put some additional hyperpriors on them, and we'll try to infer them from the data. Okay, so this will be taken care of by the data rather than the user. Okay, so we don't have to guess what are their value because the data will tell us what are their values. Okay, so I just want to quickly mention that, so for all the talk today, I'm using this binary formulation because it's super easy to explain because it's either yes or no type of question, okay? But in genetics, we can go to even more complicated situations where this function annotation can be continuous. So here's an example illustrating that, but it's not going to be the focus today. So in short, in this example, we recently illustrate that we can utilize the topology of a given gene regulatory network to come up with some kind of continuous score. And that kind of continuous score can help us to annotate the function of each SNP when we analyze those SNP in GWAS. So this work can be a talk by itself. So I'm not going to talk too much right now, but I'm happy to chat more offline. Okay, so back to our main course. So today we're working with the binary annotation. Even more specifically, so actually this binary annotation, I've tried different versions like enhancer, like chromatin loops, like tissue-specific gene set. But today I'm just going to focus on the simplest case where we're looking at the gene set. Okay, so if SNPJ belongs to any gene that is part of an important gene set with certain biological function, I'm creating the corresponding binary annotation AJ. So all I have to do is to combine the RSS likelihood that I presented earlier that input the GWAS summary statistic, okay? And then combine this likelihood with the annotation dependent prior that I just go over. Okay, so beta J will be having different prior distribution depending on whether SNPJ belong to this gene set or not. So you can see the whole thing, the whole framework is working because of beta serves as a bridge. Okay, so beta shows up in the likelihood part as the unknown parameter, and then beta shows up again in the prior specification. And then by using Bayes' theorem, I can easily connect this likelihood with the prior. Okay, and that is how I utilize the GWAS data to estimate all these unknown parameters showing up in this Bayesian hierarchical model. So I will talk about two applications based on this framework. So the one application is called gene set enrichment analysis, and I will be formulating this as a Bayesian model comparison problem by comparing the enrichment model where any of the two enrichment parameters is non-zero with the baseline model where both of the enrichment parameters are exactly zero. And to perform the comparison and calculating a Bayes factor measuring the evidence for the enrichment model against the baseline model. And if I'm getting a larger Bayes factor value, that's a strong indication that the data shows more support to the enrichment model compared to the baseline model. And here's an example of applying this to a real GWAS of the low density lipoprotein or LDL cholesterol level published in 2010. And then the input gene set that I utilize here to create a binary annotation come from 17 protein coding genes that have important functions in lipid transport. And then my annotation is binary. Basically, is the SNP falls, so is the SNP falling inside the gene set of the 17 gene, yes or no? And then after I input the data into my software, I produce a Bayes factor for enrichment of this gene set as 10 to the power of 65. So that is substantial evidence supporting the enrichment model for this particular example, which makes sense because the gene set I'm choosing here are all responsible for lipid transport, and then the phenotype I'm looking at here is also a lipid phenotype. Okay, so this serves as some kind of positive control in real data to get a sense whether this method works or not. But other than the biological knowledge to support the conclusion here, I also want to look at the data without utilizing any statistical model to get some additional verification. So this is what I did. I simply visualized the GWAS summary statistic for two groups of SNPs. So you can see there's a red distribution here. This corresponds to the distribution of the GWAS score for the SNPs outside the gene set. And then this green curve corresponds to the distribution of the distribution of z-score for SNP inside the gene set. So the red distribution is very concentrated near 0, whereas the green curve is a lot more spread out and often show, like from this heavy tail, we can see it often show a lot of very large z-score in magnitude. So by just doing this simple data visualization also supports our enrichment discovery, because this green distribution clearly has a lot more SNPs with very large z-score compared to the red distribution. Okay, so the second application of this Bayesian hierarchical framework is to prioritize genes that are likely associated with a given GWAS phenotype. So we simply calculate the posterior probability that at least one SNP in a gene G is associated with the trait using this formula. So actually for every single application I presented earlier, I perform a lot of simulations to verify things are working, but I didn't show them. So here I'm showing the only simulation result today. So I basically simulate my data from real world genotype data. And then because I simulate the GWAS data from the real genotype myself, so I know the truth, that is, I know which gene are the true the causal gene. I can even say a strong word like causal, that are responsible for the phenotype. And then I utilize my method and other people's method to identify these true genes. And clearly our method is doing a great job picking up those true genes and not making, and very competitive compared with several other methods. Just like the prediction part, we also have some external evaluation that has nothing to do with me. So this is a study published 2 years later after my study. So this group of people, I think, is in Brown University. So they reach out, and once again, I teach them how to use my software, and then that is all. They apply my software to their own simulation. And once again, our method is not the best anymore because they developed their new method, which is the best in this paper, but it's still very competitive. So if some of you know, MAGMA and SCAD are very popular methods in the field of identify genes associations. And our approach called RSS, this black curve is actually outperforming the MAGMA curve and also the SCAD curve. That is really reassuring that our method is still very competitive even 2 years later after publication. So there's a very nice feature about our method. So we have two applications, right? So one is gene set enrichment and the other is the gene association. So actually the gene set enrichment can inform the gene association in a very natural way. So here's the same example of the LDL GWAS LDL GWAS and the 17 genes relate to the lipid transport. So I have very strong enrichment here, okay? And then for each gene, I calculate two posterior association probability under two different models. So we have this enrichment P1, so that's evaluated under the enrichment model, and then we have the baseline P1 that's evaluated under the baseline model. So these are the results. So the y-axis is the enrichment P1 and the x-axis is the baseline P1. So you can see there's a group of genes here. That's actually the easy cases. So with or without using the pathway or the gene set information, just by looking at the GWAS data alone, we can identify them with very strong confidence, okay? The posterior probability is nearly 1. However, there's another group of genes that if you only look at the GWAS data alone, you have very weak evidence about association. But if you introduce the gene set enrichment information into their analysis, their posterior association probably all the way rise to very large value, almost like 0.9, 0.95. Okay, so this kind of potential gain of power really comes from the enrichment, because without the enrichment information, these genes will not be nominated in our analysis. And among these interesting examples, one gene caught my attention. It's called MTTP. So first of all, this gene is part of the 17 genes regulating the lipid transport pathway, so it's supposed to be important. However, this gene was not identified if we only consider the GWAS data. So the baseline P1 is very small, 0.14. But after we introduced the enrichment information, the evidence increased all the way to 0.99. Okay, identify this gene without any trouble. This gene caught my attention because I happen to know that this gene has known functions in regulating lipid metabolism according to multiple experiments in both mice and humans. And furthermore, this gene is actually a potential drug target to lower LDL levels, and that's exactly the same phenotype we're looking at in GWAS. Okay, so speaking of GWAS, let's see what kind of a job GWAS can do in terms of identifying this gene. So first of all, the result we're producing here come from the GWAS of LDL in 2010. And then in the same GWAS, this gene was not identified by the standard single SNP association testing part, okay? So the p-value is too large to call significant. And then later on, people increased the sample size. I think they doubled the sample size. So from about 100,000 to nearly 200,000. Once again, the p-value is not strong enough to call association reliably. It is until very recent when we published the largest GWAS of LDL involving 1.65 million people a few years ago. So you can see now we're reaching 1 million sample size, but now we're able to identify this gene with very significant association using the standard GWAS approach. So this somehow confirmed our previous computational finding based on a much larger sample size. But moreover, this illustrates that our approach gives people some cost-effective option of identifying additional disease-causing gene if they don't have the luxury of increasing the sample size as we did several years ago. So hopefully I have convinced you that the RSS framework, the regression with summary statistic framework, can be useful in at least 4 different applications. So there are several more. There's like fine mapping, there's like a Mendelian randomization that I don't want to get into today because there's a lot of material presented. But I do want to conclude today's talk with some brief overview of what I'm doing right now to expand this line of research in order to address the emergent challenge associated with this widely available summary data. So actually, there are 3 directions I'm actively pursuing now about genetic interaction, population diversity, and also privacy protection. But for the interest of time, I will just focus on the genetic interaction. So genetic interaction is an important thing to study in genetics in general, but here I'm specifically looking at a type of that type of interaction that only involves enhancers, which is a non-coding element in the human genome. So over the past few years, I have the luxury working with people at Stanford and Fred Hutch Cancer Center. So they introduced me to this very fascinating experimental system where we can either perturb a single enhancer at a time, or we perturb 2 different enhancers for the same gene at the same time in the same cell line. Okay, so based on the so this is a figure illustrating applying this kind of experiment to a famous oncogene called MYC. So based on this type of experiment, we can further develop machine learning algorithm to infer how enhancers are interacting with each other in a network. And this is an example of deriving this network for the same oncogene called MYC using the experimental data from cancer cell line for B cell, okay? And because this network comes from the B cell lines, I'm combining this network with 2 GWAS data related to B cell. So one is leukemia and the other is Crohn's disease. So long story short, so we're able to identify very significant genetic interaction here in the sense that I take one SNP from one enhancer, and then I take another SNP from another enhancer that forms a very strong functional interaction inside this network. I use the GWAS data evaluating the individual odds ratio or effect size for these 2 SNP in 2 different enhancers. And then I can also get to evaluate the joint effect size or joint odds ratio of these 2 SNPs simultaneously. And this allows me to identify the genetic interaction effect. So here we can see the interaction effect is a lot larger than the individual odds ratio derived for each SNP alone. And we observe the same pattern for the other Crohn's disease. So these initial findings really got me thinking that, okay, now maybe it's with this kind of experimental data become more and more available, it is the time to seriously investigate genetic interaction in the context of GWAS, because for people who are familiar with the GWAS literature, genetic interaction has been overlooked for at least 2 decades, okay? So our approach is very simple. We simply introduce a interaction term into the GWAS data, into the individual level data model for the GWAS, and then we introduce a corresponding effect to estimate for this interaction term. So for this interaction, if we construct an interaction for any combination of SNP in the genome, that's impossible. We don't have the sample size to infer that huge search space. So here we're using the enhancer network to narrow our search. So we're only looking at the genetic interaction coming from this kind of enhancer interaction, which is a lot more smaller than the entire genome in terms of searching this kind of interactions. And then I'm very grateful for this longtime colleague, Dr. Xueqiu Lin at Fred and Hatch. So her lab has been producing this type of experimental data so that we can utilize to define the interaction here in a more efficient way. So once we have this data likelihood, and suppose we estimate these two, so we have additive effect here as usual, and then we have the interaction effect. And suppose we have these two effects estimated from the data, then we can pursue one interesting application called PRS, or polygenic risk score. So that's essentially some kind of prediction. So if you just look at this term, so this is essentially what I showed many slides ago. We're doing this kind of prediction of phenotype from the genotype alone. So this is the standard PRS approach by just considering the additive effect. So now we're pushing the boundary by including the interaction effect. Okay, and it is our belief, and we're still collecting data and producing results to prove that belief, but it is our firm belief that adding this additional term, we will be significantly improving the overall prediction performance compared to this standard approach by just looking at the additive effect alone. So now here begs the question, how can you add? So earlier we see from the RSS application, so there's a way of estimating beta A from the summary data. Okay, so that's very convenient. That's doable these days. Can we do the same thing for the interaction effect? And the answer is yes. So we can actually modify the way how we set up RSS in a way that now we can estimate both additive and interaction effect simultaneously from the summary data. So everything remains the same. There's only one modification here. So when we're making the assumption before the likelihood derivation, we need to assume that now our phenotype data are generated by both additive effect and interaction effect in the same time, okay? So this is the only modification we need. So under this new generative model, we can use essentially the same proving argument I presented earlier to derive a new interaction-aware RSS likelihood. So you can see we still have this part showing up for the additive effect, and then this is the only new component. But this new component allows us to estimate this interaction effect. And of course, I'm a Bayesian statistician, so I'm going to use Bayesian approach to estimate this additive effect and interaction effect simultaneously from the summary data by using some kind of prior distribution, okay? So these are very new work. I'm still producing results for it. So I hope to share some update with you in a different seminar in the future. Okay, so that's going to be all my material today. Thank you so much for your time and your patience. And then these are relevant papers. Some of them I talked about, some of them I didn't, but still some kind of relevant work under the overall RSS framework. And these are the financial support I have been receiving over the past few years. Without this support, I cannot produce this talk today. So thank you for the founders. And that's all for today. And I'm happy to take questions. And I think there should be some questions in the chat already.
Yeah, not yet. So
okay, that's good.
thanks, yeah. So it was a great talk. I mean, I really like the pace of the talk. I think you got into the details, which was hard because you have statisticians and people from biology background, so you had to balance both. So thank you. If anyone has questions for Xian, you can just unmute yourself at this point and ask, I think.
Okay, I'll just go back to the summary page so people remembers what I've talked about.
So I have a quick question.
Sure.
How is the entire system? I mean, how is the computational cost in all these things? Like, how long does it usually take for you to? I mean, I assume that depends on the size of the data and all that things.
Yeah.
But how long? I mean, can you give us a rough idea about the process?
Sure, I think so. So just to answer that question, so the overall cost is mainly
Depends.
controlled by this P. Okay.
Yeah.
So I've working with different P, but the usual scale of P I've been working with is about 1 million.
Yeah.
So I can maybe for this LDL example is pretty good. So for this LDL example, you can see okay. Also depends on how many gene sets you are looking at. So if you just analyzing 1 or 2 gene sets, you can get a result ready like in less than an hour. But here's the thing. I choose this gene set because, somehow I already know the answer. I just want to do some kind of validation. But in practice, we usually don't know what gene sets are truly relevant to the disease. We want to make some scientific discovery. That means we want to include a lot of unknown gene sets into the process, and we let the data speak, right? And we let the data tell us, okay, is this gene set relevant to that disease? And that can lead to novel discovery. So in that case, we need to analyze like thousands of gene sets in one study. But so I think if we have access to those HPC like we have here at Penn State, then it's not a big deal. We can just submit those jobs to run in parallel. So that's going to be still assuming we have access to those resources. Then again, like in one afternoon, we should get all the results ready for thousands of gene sets. And for 1 or 2 gene sets, you can get a result ready in less than an hour. That's actually an overestimate, but let's say over about an hour for 1 gene set. Yeah.
So is the methods publicly available for someone to use, Xian, or?
Yeah, it is publicly available. So it's in the, I didn't put it here. It's in the I think I'm using the same GitHub to share the code. So you just go to this annals paper, which is my first paper in this RSS research. I think the links they're still valid for people to download. So actually sharing software is part of the story, right? Because if we don't share the software, then other people cannot do this in external evaluation for me. And I was really appreciate for their effort because they're basically doing this, but helping me make my point. But I think it's essential having some other people proving that the tool is working without your own involvement as the developer.
I have a question, Sean. Thank you for the great talk. So with the data, if they are too large, they will be distributed in different computers.
I see. So okay. So I think so actually the P can be large, but we do have this very nice feature here to take advantage of. So I think you're thinking about, okay, it's a P by P, it's a P-dimensional Gaussian distribution, right? This huge correlation matrix, but actually it's yes, the mathematical formula is P by P, but it's actually a lot smaller than that. So in my case, I never had to distribute my data into different machines. I think if that's the case, then there will be a lot more engineering work for me to do, but I don't know how to do that. So right now, I can get all the computation done with one machine, like not a typical laptop or a desktop in the office, but like a typical, like one I think what's they call like a core or one. So basically one machine in our Penn State raw server, they're like one machine but has like a large memory and like 30 CPUs there. I think a setup like that can handle the current calculation I'm working with every day. Yeah.
Great.
Yeah.
Thank you.
Okay.
All right.
Ready to.
We're almost at the time, but are there any other questions? All right. If not, well, Xiang is at Penn State, so anyone can contact directly.
Exactly.
Xiang.
I was about to say, yeah, I'm local, so yeah. I'm
All right.
local at the University Park. In case some people are in Hershey campus, then that requires some remote connection to collaborate. But here I'm at Penn State, so if you have any GWAS related question, feel free to give me an email and we can find time to chat.
Well, thank you very much, Xiang, again for the great talk.
Thank you, Prabhani. Thank you, Musak. Thank you so much
Bye-bye.
for organizing this series.
Thank you so
Bye.
much. Bye-bye.
Bye.